I need to, all right, just one more second. And if you can tell me if you're able to see what I'm displaying, it should be the title slide.
Yes, it's okay.
All right. Well, thank you very much for having me. I need to remove something from the screen, and I'm not able to do so, but we will do our best and make it work. So again, this is really a privilege for me to be able to join you today. I really enjoyed the first sessions that I heard. Professor Ambrose Wonkam, of course, I remember when we first met, and it was with you and the late Professor Bongani Mayosi, when Dr. Gary Gibbons, who was the director of the National Heart, Lung, and Blood Institute, was visiting in Cape Town. So it's really been a great pleasure. Even though you're much closer to NIH now with your location in Baltimore, I'm hoping that we get the opportunity to connect more. But truthfully, these sessions have been very, very inspirational. Hearing the progress that you've made, especially the career progression of the trainees that you've had, and the big ambitions you have from going from Africa to global. This is all music to our ears. So thank you very much for the invitation, for the update, and for the excitement that you bring to the research on sickle cell disease. Let me also say that I bring you greetings from Dr. Gibbons, who's director of the NHLBI, from Dr. Julie Panepinto, who is the division director for Division of Blood Diseases and Resources, and certainly from the entire leadership and staff of the Center for Translation Research and Implementation Science, which is the center I lead. So it's really a great pleasure. I also want to thank Professor Julie Makani, who called me personally and encouraged me to accept the invitation, and I'm really delighted that I did. I should tell you any conflicts in the presentation of this keynote lecture. And even though I am here and doing this as part of my official duties at NIH, I should also tell you that nothing I say here this morning should be seen as representing an official position of the NIH or the United States government. Let me also begin really by thanking all of you, this being your 11th Sickle in Africa Consortium meeting, the 10 previous successful ones you've had, all together really make us very, very proud of this program. I want to make sure I extend the gratitude and appreciation from the NIH and from the NHLBI to our co-chairs, our clinical coordinating center leadership, our data coordinating center leadership, and all of you PIs, your co-PIs, and especially the patient participants who make this research possible. So really congratulations on what you've done. And I also have to tell you, we have great expectations of what you are about to do. And frankly, I should also highlight that I'm not here to lecture you on anything that you do not know already. In fact, I'm here to remind all of us, both you and us at NIH, about the critical role of the research that you're planning, especially how you expand and refine the registry phase of this work and the potential transformative impact that the work that you're doing can have on the lives of children and adolescents and adults of all ages in Africa. And as Ambrose mentioned, the learnings that you help us identify and surface can have real impact in India and the rest of the world. So again, thank you very much. I have only 4 main objectives that I would like to accomplish in this lecture. As you all know, translational research means different things to different people at different times. So it's important to begin with some clarifying principles and the definitions that I'll be using throughout this lecture. The second and third objectives highlight some of the really important research advances we've had over the last decade, a decade during which the Center for Translation Research and Implementation Science has been in existence, but also identify many of the challenges, the impediments, and the barriers that stand in the way of taking all the research findings you have and translating them into practice. And finally, I'd like to be able to highlight the very crucial role of implementation science, dissemination research, and the role that plays in turning these discoveries into health impact. Back in 2013, the year before CITRIS was established, we drew this cartoon to reflect the entire translational research pathway from basic science and bench research through bedside research to patient trials, particularly phase 2 and phase 3 clinical trials, all the way to the biomedical and socio-ecological model that reflect real-world research turning discoveries into impact. The T1, T2, T3, and T4 phases, as you know, don't have very, very sharp boundaries, but we clarify that we recognize there's an early stage translational research, which really deals with bench to bedside, and there is a late stage translational research, which is where CITRIS was going to focus the real emphasis on making sure that there's real world routine application of the research that we invest in. That year, the Institute of Medicine, which is now the National Academy of Medicine in the United States, put out what they call the operational phases of the translational research, identifying also what I'm showing you first, the translation from basic science to human studies, emphasis on basic science, early translation, proof of concept in phase 1 clinical trials, and also phase 2 and 3 clinical trials. But they also added that second phase, that later stage translation was important. And if you look at the very bottom, and I'm sorry, my mouse pointer is not showing, but you can see that the Institute of Medicine identified the T4 phase as what represented true benefit to society. I think this is very important. And for us, the message is, it's great if we're doing wonderful basic science research, we're doing innovative early translational research, but if we don't carry it through to make sure that those research findings are benefiting the communities we serve at the street level, on a day-to-day level, then we really have not identified and highlighted the true benefit to society. So as a quick definition, early translational research reflects how we turn fundamental discoveries at the basic science level to real impact, to human studies, identifying efficacious interventions, effective interventions that set the stage for real implementation. The core principles, as I mentioned, involve the emphasis that we put on rigorous bench to bedside approaches. Just because the different T phases, T0, T1, T2, reflect different areas of expertise, it becomes very important that we bring together a real consortium of people with very diverse, multidisciplinary backgrounds, and also partners from across many sectors. But the importance that we put on regulatory, safety, and ethical considerations are all very, very important in translational research. This is just another cartoon from NIH highlighting the important phases, and I regret my mouse pointer is not showing, but you can tell that there's an emphasis on preclinical and animal studies in the early phase. You go through first in human studies, proof of concept studies, and a strong focus on efficacy and safety. Now, implementation science, on the other hand, is the scientific study of the methods, the strategies that we use that can promote the uptake, the adoption, and the integration of new evidence-based interventions into our routine clinical practice and policy development. So it really takes us from the fundamental discoveries, the first-in-human studies, to safety and regulatory ethical issues, but we need to make sure that we get down to the real world level so that we can improve quality healthcare and health for all. Some of the core principles have to do with sustainment of the evidence-based interventions that are adopted, the strategies that are adopted, the real use and emphasis of validated conceptual and theoretical models, frameworks that have been proven to work in different settings, and use these to systematically plan, to monitor, to evaluate, and assess the barriers as well as the facilitators that drive uptake or really delay uptake, and be able to monitor this over time. So the context of the research, the context of the community setting becomes very, very important. Multiple partners become very important. The need to engage experts across different sectors become important. And most importantly, we put an emphasis on, are these new interventions, devices, drugs that we talk about affordable? Are they acceptable within the cultural context? Would people actually take them when we have them available? And can we sustain them? And if we're going to be able to do all that, what is the research that we do to advance that? And I'm here to tell you that the National Heart, Lung, and Blood Institute is committed to supporting implementation science and implementation research that ensures that the fundamental discoveries you make, the early translational research that you, discoveries that you find, can all be really turned into real impact. The good news is the Sickle in Africa Consortium is already paying attention and putting an emphasis on this. In fact, nearly exactly a year up to considering this week, that's when the consortium put together this webinar that Dr. Bruno Mbando moderated, and a session that was delivered by Professor Emmanuel Pepra from the NYU School of Global Public Health. And I'm very proud to say Dr. Pepra was from the CITRIS, our Center for Translational Research and Implementation Science, before he went to head the studies there. But it's really good to know that in addition to all the wonderful things that you're talking about, as you advance your registries, you are also paying attention to the core principles of implementation science so that the research discoveries you make can really have impact across Africa. And as Professor Ambrose Wonkam mentioned, that we can begin to have impact at the global level as well. Another good news is compared to 10, 15 years ago, we now have several resources, as you see on the right, that are textbooks and major publications that focus on implementation science. In fact, in the very bottom right, I show you over the last decade what we now have dedicated journals for implementation science, as you see here, as well as implementation science communication. So there is good news, and I'm hoping that we all pay attention and leverage dissemination and implementation research to really make sure that all the other research we do can really, truly benefit society. Multiple institutes and centers at NIH also invest and commit to implementation science. And I share with you one example of a resource, this one put together by the National Cancer Institute. But the core principles are the same for cancer, they're the same for heart disease, they're the same for sickle cell disease. And so this is freely available, and I encourage you, when you get the slides, the hyperlink at the bottom will take you to the site and you can download it. The Fogarty International Center, that also supports many of you and the research you do, also has a strong commitment on advancing implementation science. And so again, I encourage you to visit the appropriate sites for the different institutes and centers, including our own, so that you can take advantage of the resources available. Now, in the next segment, I want to address some of the research advances that we've seen over the past decade and the impact that they have on improving care for individuals with sickle cell disease. All of you are very much aware of the work of James Herrick from Chicago, although the original discovery was made by one of his interns, who then reported to James Herrick, and Herrick, of course, got all the credit for it almost 115 years ago. But since then, we've also had several major advances, including the work from Nobel laureate Linus Pauling, and these two citations that I have on the bottom right in the Bulletin of the New York Academy of Medicine back in 1964, as well as Ingram's paper in 1958. But in the last decade, actually, we've even had more impressive advances. But what all of us would agree on is it took too long since sickle cell disease carried the label of molecular disease, since the original descriptions of Herrick, since the work of Linus Pauling, and almost everything else that came after. It took way too long to really make the significant improvements that we're beginning to see in the last decade. So great excitement, but we still have a lot of work to do. These advances we've seen have improved screening, they've improved diagnostic testing techniques. Stem cell transplantation is one example, and of course, most recently, gene therapy and gene editing strategies, but certainly the development of biomarkers that enable us to predict and identify high-risk individuals, as well as very novel strategies that leverage newer innovative mechanisms, such as the use of anti-sickling agents, fetal hemoglobin inducers, as well as other adhesion molecules and novel discoveries from vascular biology applied in sickle cell disease. The wonderful thing that all of this is not restricted to hematology, but we can bring in advances from cardiology, from vascular biology, from metabolic science, really unique opportunity for us to make all of these discoveries work in a day-to-day level for the communities and the patients that we serve. You are all aware of these manifestations that I show on the right, but the manifestations and reflections of the advances in translational research that we've been able to make. This is a single slide that shows, even within the last 10 years, just the last 3 to 5 years, several of the FDA-approved treatments for sickle cell disease that were not available 10, 15 years ago. But the real question is, how affordable are these? How acceptable are these? What are the facilitators that would enable us to get most of these to as many people as who need them, so that people who live in cities as well as villages benefit equally, people who live in rural areas as well as urban areas benefit equally, and that people don't go bankrupt, making themselves available and taking advantage of the science and the discoveries that we've made. That is where implementation science becomes important. If there's one figure that has impressed me and improved my own understanding of the advances that we've made, is John Tisdale's figure published in Science 3 or 4 years ago that looks at the current and the future treatment, the excitement about what is possible, but also what we know works today. that we need to do everything possible. And I'm hoping your registry studies, particularly done in a randomized, pragmatic approach, can really help make better use of the treatments we have today, such as hydroxyurea and bone marrow stem cell transplantation, as well as we think of the future treatments, particularly ex vivo gene therapy, taking advantage of genetically modified stem cells, using either gene addition or gene editing, but also in vivo gene therapy. So the horizon looks great. But as we contemplate the future, we should also learn from the lessons from the past. Some of the major challenges that we still have with the administration and sustained use of hydroxyurea. What convinces us that the future wonderful drugs we develop will not suffer the same fate? It's up to us to make sure that we do the research that enables us to identify the supports, the tools, the techniques that we need to ensure sustained use of proven effective interventions. We don't need to look very far to find examples of proven effective strategies that can be applied today. Yes, the future looks great, and the wonderful things that we can have are all very, very exciting. But I hope we can also pay attention to proven effective strategies today that are available in Angola, in Uganda, in Kenya, in the DRC. And as we look towards the rest of the world, how we spread and disseminate this information. Again, the example from REACH in the use of dose escalation and optimization of hydroxyurea treatment in children is really very, very instructive. Here showing the profile of the dose escalation use in the different sites. But for me, the most important findings, as I'm sure Professor Chusolo would share with us, is that the compelling evidence of the reduction in painful events for patients, the reduction in the frequency of acute chest syndrome, as you see on the right, again, reductions in the incidence of malarial and non-malarial infections in children in Africa. And finally, and very impressively, the significant reductions in serious adverse events on the left and death on the right. The challenge for us is, how do we make sure that every child in Africa who has sickle cell disease can also benefit from these findings? How do we make sure that the hydroxyurea that can deliver these remarkable findings is affordable, is acceptable, is sustainable, and then we can make sure that we continue to learn and develop better strategies for really making sure that people benefit. These are examples of the challenges that we face, and I'll highlight a few. About 21 years ago, and I'm showing you the picture of Claude L'Enfant on the left. Dr. L'Enfant was the NHLBI director from 1982 to 2003. He was the longest serving NHLBI director we've ever had. And when he had the opportunity to give the keynote lecture, which was then called the Shattuck Lecture in Boston back in 2003, he sort of shocked everyone by saying that the millions and millions of dollars that we've put and invested in research, particularly clinical research, with the hope that we can turn our research findings into clinical practice, most of those findings are lost in translation. And he emphasized that it was not just recent research results that are not finding their way into clinical practice and public health behaviors. There's plenty of evidence that old research outcomes have also been lost in translation as well. That was 21 years ago, and that statement is still true today. Much of our discoveries about the importance and the benefits of hydroxyurea are still lost in translation. So how do we turn this around? If you look at several of the challenges particularly in new drug development, new device and new technology development, there are two valleys of death that are described in the literature. So we have an initial value of death where a wonderful discovery, and again, I really regret the pointer is not showing, and I'm not sure how to address that. But if you look at the image on the right, research done at the bench in these beakers and tubes in university and research institutes often identify highly efficacious drugs or techniques, but then they meet important hurdles and they have difficulty obtaining financing for commercialization. It leads to a rapid death of the drugs and devices and molecules that are identified. I'm here to tell you that the NHLBI has a platform that supports novel, creative ideas to enable them to gain the appropriate financing and become commercially viable so that they can proceed. So even when you look at the first value of death, we have strategies at our institute to support investigators to overcome that barrier. But even among those who overcome that first barrier, there's a second barrier where you have a highly efficacious drug, you have proven its commercial viability, but then you lack the appropriate funding to do the expensive human clinical trials to demonstrate clinical effectiveness, and therefore it suffers a second death, that second value of death. Within our institute, we prioritize clinical research, translational research, implementation research that enables us to do the appropriate studies that can move these novel discoveries, novel drugs, and novel strategies forward. And I'm hoping that you hear me say that we are interested in your investigator-initiated ideas that can get the research support that we have. It's unfortunate that we can't support everything that we receive every year. We have outstanding applications that we are not able to support. But you should never let that let you down, because there are other funding agencies that you're able to follow through. But the essential message is investigator-initiated research ideas are very, very welcome. And we hope you use the work that you're doing in Africa, the registries that you're advancing and enhancing and expanding, and very creative and innovative ways to help us overcome the barriers and the challenges for making sure that research discoveries get to people on the street, get to people in the communities that we serve. This is a more recent paper actually published earlier this month, Julie McCarney and colleagues, showing the multiple instances where we face translational gaps, translational impediments, especially at the intersection of the interactions between academia and government and the private sector, whether it's in making sure that we have an appropriate workforce to do this research and be able to move this forward, whether we have in-country technologies that are supported in order to enable us to do this important work that we do, how we protect intellectual property in the countries in Africa where this work is being done. Within the government arena, the importance of national priorities aligning with the direction that the science is going so that we get the appropriate level of support from budgetary allocations, and even within the private sector, ensuring that the drugs and the devices and techniques and technologies that we develop are safe, they're available, and they are affordable. So there are multiple challenges, but the important message is, as you think of all the wonderful research that you want NIH to support, that you want to be able to do, that we also think of the barriers, the impediments, and be able to bring in the key stakeholders who, especially in low and middle income countries, who can help us make sure that this work makes progress. I show you the example of the unique strategic partnerships we've put together on the path to cures for sickle cell disease. And again, at the bottom right, you see the very, very important role that this consortium plays in that effort. But the slide is to highlight that we can't do this work alone. and we need to identify strategic partners that we bring into the fold every step of the way, including meaningful engagement of patients, patient advocates, their families, and the general public in making sure that this research is supported, is sustained, and we find ways to translate the findings into real impact at the street level. In my final segment, I want to focus on implementation science and also bring you the message that at our institute, at the National Heart, Lung, and Blood Institute, implementation science is one of our core enduring principles. So every time you're putting forward an application, whether it's in core basic science or early translational research or late stage translational research, thinking about the challenges and the barriers that your research findings would face when you develop and publish your results really helps us know that you're thinking way ahead, and we have a much, much better chance that your research findings would get translated into real world thinking. So there are several strategies that I would mention, and I hope that all of you have been thinking about these, but the time for just focusing on fundamental research just for its sake, and the science of early and late translational research just for the sake of science, that time has come and gone. We need every step of the way be thinking about the patients that we're trying to serve, the communities we're trying to serve, the barriers they face, and use the core principles of implementation research to identify those barriers, those facilitators, so that the research you do and the findings you get have a much, much better chance of being implemented in the real world. Now, establishing that a new drug or a new technique, a new technology is efficacious and effective is important, but it is really not enough, and it does not guarantee that it would be uptaken into routine practice. So we really need to explicitly study the implementation of the innovation. I think up until now, we've spent a lot of effort, and I'm not saying you, I'm saying all of us in the biomedical field at NIH, at NHLBI, we put a lot of emphasis on the innovation and the science and the creativity, and very often we think about how it will be implemented in the real world at the very, very tail end. My essential message is we should be thinking about implementation right at the beginning, and we should explicitly study the new innovation we're thinking about introducing, or the new way of using an old drug, and how the communities we serve would actually trust us, how they would believe what we say, how they would accept what we teaching and professing and sharing, so that they can really benefit. We clearly must identify what the barriers and the facilitators are that stand in the way of sustained uptake. And these barriers vary. It may vary from Bamako to Accra to Abuja, but this is where implementation science become important. You are the experts of your communities. We want to hear from you how you'll be tackling those barriers and how those barriers are going to make a difference, how the facilitators you identify and the solutions for the barriers can really make a difference, so that every dollar we spend on the research can have remarkable return of investment. Why? Because the findings would not just be published in journals and sit on bookshelves, or these days sit on mega drives, but actually have real world importance and implication. We must also spend time to study the context within which any new innovation, any new drug for sickle cell disease is being addressed. How would that context and what we learn about that context affect the acceptance, the adoption or adaptation, and also ensure that it's used with high fidelity? These are all core principles of implementation science. We have to assess not just cost of the drugs, but would that cost be affordable by the community for whom we're proposing this? And if they have the resources and the interventions and drugs and devices and tools and techniques are affordable, are they even available in the villages and small towns as they may be available in the major cities? access is important, and always thinking about how we can optimize and maximize broad reach of the communities we serve, not just the patients who come to us at our major tertiary care hospitals and medical centers, but also people who are working with community health workers, village health workers, social scientists, social workers. All of us, we need to find ways to make sure that the science we support is readily available, can be adopted, can be practiced, can be sustained, and lead to real benefits and improvement in quality of life and longevity of life for our communities. Finally, I think it's really important, and we are very, very interested in research applications that help us understand why specific drugs or strategies or techniques work. First, which ones work and how they work, and learn from also the ones that don't work. All of these come together under the broad rubric of implementation science, whether it's dissemination research or implementation research. This is something that the time has come for us to emphasize, so that the wonderful research that you propose, when your findings have impact, can really have sustained impact, not just in Africa, but globally. This is a cartoon that I put together that for me helps me explain why implementation science is important and where the barriers and facilitators come in turning our discoveries into help. If you look at the very left and the bottom left, those circles represent the different innovations, and they could include evidence-based proven effective drugs, the tools and techniques, and the different strategies. But as we think about research in this arena, we always have to think about the individual patient at the very center and base of those concentric colored circles in the top left. Is the individual, their families and their friends, and the communities in which they live, but also importantly, their healthcare providers, their doctors and nurses, and social workers and community health workers, and the health system within which they operate, the social and environmental aspects, the community resources that make certain approaches feasible, but other approaches not even practical. All of these are important. And I think we are calling on all of you that the research that we do must take all of these into account, because by doing that, we can absolutely engage the implementation frontier, the barriers that we must address so that our research findings can be successful. There are drugs that are underused. Hydroxyurea is key. There are certain techniques, drugs, and devices that are also overused. But in general, there's a clear mismatch when it comes to the evidence that we know and the practice that we see on the street. And that is what implementation science is trying to address. When we're looking at implementation science in sickle cell disease, we're not talking of levels of oxygen saturation, the degree of sickling, the degree of pain, but we're talking about, are the strategies and the research discoveries we're making acceptable? And acceptable put within a cultural and linguistic context. If we don't study that, then the chances that our findings are going to benefit people from across different spheres of cultural, social, environmental abilities becomes very, very limited. So implementation outcomes are unique. Not only must they be acceptable, but are they appropriate for the setting? Newborn screening is definitely appropriate for the general population and in adults. But specific strategies may have real appropriateness and acceptability in one setting, but not another. What defines those two areas? How can we use the registries that we're developing to help us tease those out, but also have real, rigorous, randomized data from the registries that can inform policy, can inform funding, and really lead to improvement? All we're trying to do is the image you see on the far right in green. That's we want to increase the years of life, but not just longevity. We also want to improve the quality of life and remove disparities that we see between urban and rural areas, between people across different socioeconomic status. Health must come first. And by thinking of implementation science, we have a much, much better opportunity to really highlight all those metrics at the end. Implementation outcomes are not biological variables. We have multiple strategies that we know, if you put in place, can improve quality of care for sickle cell disease. But how many people are we reaching? And how can we improve that reach? Of the people we reach, how many of them use their doctors or follow their doctor's instructions with high fidelity? But what are the barriers to that fidelity? Is it education? Is it literacy? Is it trust? We have a responsibility to study all of those factors so that the research we do can really have sustained impact across the globe. Another cartoon that also helps me is what we're trying to do is begin at the left in red is advance fundamental discoveries. That's what NIH, the single largest funder of biomedical research in the world, that's what we do. And that's why the NIH tagline is turning discovery into health. But that process is not as linear as I've shown in the image. Going from fundamental discovery to health impact in the community is complex, and it faces multiple barriers at multiple levels. If you look at what I described as an early stage preclinical research, you have the first valley of death and the second valley of death going from first in human studies in phase 1, phase 2, and phase 3 trials to how we design comparative effectiveness research, how we do efficacy and effectiveness research, and dissemination and implementation research. But even when we are successful in going through all of these phases to get to real health impact, we have a major barrier of clinical inertia and policy inertia. How do we tackle those? Doctors who know that hydroxyurea is effective and still don't prescribe it, or when we prescribe it, we don't have the resources in the community to ensure that everyone benefits. The clinical inertia, the health policy inertia, the challenges that ministries of health face, all of these must be brought together for the research that you are doing to have the greatest impact. A few other techniques that I want to mention because we are very, very interested in supporting implementation research, but there are some types of research that really we don't get very excited about. Why? Because the traditional approach of first designing efficacy studies, taking 5 to 7 years to complete the studies, publish the studies, and then use the findings to design new effectiveness studies, do 4 or 5 years to complete the effectiveness studies, publish the results, and then based on the results, now design implementation strategies so that we can implement what we find. It takes too long. And that's why it is not a surprise to anybody that on average, proven effective interventions take about 17 years for only a small fraction of the research to benefit people in the street. We need a change. And one example of a change that's very, very effective and we encourage is where you design hybrid approaches that both explore effectiveness as well as implementation at the same time. There are 3 types of hybrids. I know that Professor Pepra went over it, and all of you are familiar, but we get excited when we see clinical effectiveness studies that also have a component of addressing the implementation barriers or implementation facilitators. We know that that reduces the length of time it takes for your research findings to have impact in the real world. So give some thoughts to hybrid approaches. This is the same concept as I'm showing on the left, but I use it side by side with some of the most new approaches in the last decade, in fact, very, very recently published just last year, Justin Knox and Alvin Gang and Jeff Curran, several giants in implementation science, talk about the new approach of using the observational implementation hybrid. And I'm hoping that you all think about it as you design your registry studies. It'll be wonderful to see new approaches that take randomized registry strategies with implementation in mind. It would help us make sure that the benefit from the research you're doing now, we don't have to wait 17 years before we see the impact in Africa and the rest of the world. I'll conclude by just reminding you that the NHLBI is really committed to this work. Here are the 8 strategic objectives that our strategic vision highlights, and I've put in bold red the clinical research and implementation science as one that my center strongly supports, and we hope you will send us outstanding applications that help us make progress. These are the enduring principles of our institute. Every year, we fund at least 700 R01s. I was delighted to hear that the consortium is already planning R01 applications. You're already planning D43 applications. That's great. And I hope that some of that work also brings into play implementation research. We strongly encourage you to pursue the R01s. Very often, people want to wait and ask, when is our next funding opportunity announcement coming out? Well, let me share a secret with you. The funding opportunities constitute less than 20% of our budget. 80% of the budget we put out to support research goes to support investigator-initiated discovery research, whether it's early translational, late translational, implementation science. That's where the money is. And we encourage you to really increase the applications you send to us that really looks at strategies to benefit the communities in Africa that you serve in. You will see that enduring principle number 4 is support for implementation science that empowers our patients and our communities and enables our partners to improve health in all communities, in children, in adolescents, and adults of all ages all across Africa. Finally, I also want to mention that we are very, very interested in training and career development. We work hand in hand with the Fogarty International Center. So that's why I was delighted that you're thinking about the D43. We should also think of the individual career development of the K43s and the K01s for US partners. But certainly keep your eyes open for additional opportunities that may become available. There are lots and lots of people at NIH who are rooting for you and advocates for you. And we want to make sure that where you have questions, you reach out to us, that we can provide you the technical assistance so that your applications can be successful in getting the research. Let me conclude and save the remainder of the time for dialogue. But I made a few points that I want to emphasize. Translational research may mean different things to different people at different times, but at the end of the day, the translational research pathway is the process we use to make sure that our fundamental discoveries actually help turn into real impact in the real world. Is to turn discovery into routine, day-to-day, high-quality health and healthcare for sickle cell disease. The second point is, none of us by ourselves can do this work alone. We need a real broad, diverse coalition of multidisciplinary experts from across multiple sectors that come together, and then collectively we meaningfully engage patients, patient advocates, their families and friends, the citizen scientists, the communities that we serve. That's really essential for us to be successful in making sure that our research has impact. The third is that very safe, very efficacious, and very effective treatments are available today. We saw in the REACH study findings, the challenge is developing the strategies that would maximize how many people we reach in West Africa, in East Africa, in Central Africa, in other parts of Africa, and around the world. And to what extent are these techniques available, affordable, acceptable, and used with sustained high fidelity? That is our challenge. And we have the resources to support research that helps us identify these strategies. We know that advances in sickle cell disease have really ushered in really exciting and novel therapies with real promise of cures in the near future. These are remarkable improvements that we are likely to see in the quality of life of sickle cell disease patients. But we have to make sure that we learn from lessons of the past with hydroxyurea so that we make sure that the gene therapies that are now available would be affordable, would be acceptable, can be sustained, and can really have impact. And finally, and this should have been my most essential message, we thank you. We thank you for the work that you've done already. We thank you for the work that you're planning to do. We thank you for the creativity that these registries that you are putting together would help advance and resolve many of the challenges that we face on the continent. And we thank you for your continued commitment to helping turn scientific discoveries into high quality health and healthcare for all in Africa. Again, my center is celebrating its 10 years. And so we hope you join us in not only celebrating, but taking all the lessons we've learned in the last 10 years and help us chart a really bright future for the next 5 to 10 years as we work together. So thank you very much, Sickle in Africa, for inviting me. On behalf of NHLBI and Citrus, we wish you a wonderful next 4 days in your consortium meetings and look forward to seeing many applications from you at NIH, and we'll do the very, very best we can to support meritorious applications. Again, thank you very much, and I'll stop sharing my screen so that we can go into discussions.
Thank you very much, George Mensah. We are clapping for this great key talk. Thank you very much. And can you stay for some minutes for the discussion?
I'll be delighted. I'll be delighted to stay.
Thank you. Thank you very much. I think that your talk is a very incitative talk for all the research members who are here. And we have some questions from the audience. And my colleague, Dr. Sao, will move with the microphone. No question? I think you want to question. Yes, please.
Thank you, Professor Mensah. I wanted you to comment about the aspect of the knowledge that researchers or scientists have and how they go on to use that knowledge to fill the gap that you're describing. And this gap I'm looking at is the aspect of health equity. How can we go about that? Thank you.
Excellent. I'm sorry, I didn't get the name, but thank you very much for the question. And I'm delighted you asking that question. You know, 10, 15 years ago, after a keynote lecture like this, the first question would be about what are the mechanistic approaches and the pathways and the enzymes involved in making sure that red blood cells don't sickle. And that was wonderful. That was the type of questions and the type of knowledge that we shared. So I'm glad you're asking about equity. I think we have to admit that we may not be the best in sharing knowledge. There are people who are experts in knowledge dissemination. There are people who are experts in looking at different strategies for engaging communities and sharing knowledge based on the age group, the generation, the setting. And the important message is, as you put your research team together, make sure you have at least one person who has that expertise and can help us. In addition, over the last decade, in fact, maybe the last 5 years, we now have equity-specific implementation frameworks, right? It's one thing to ask the question, this new device, this new drug, or even this old wonderful hydroxyurea, we're going to study how we can successively leverage the rich findings and do dose escalation and optimization. But the question becomes, do you fully understand the literacy level of the communities that you're working with, the cultural importance, what the community themselves indicate is important? What are the equity challenges, the socioeconomic imbalances? If you are aware of those, how you share knowledge, all of a sudden changes. So the simple answer to your question is, it's an extremely important area of research for us. There are equity-specific implementation frameworks that you can use. They're validated and it can help you. In addition, I'll encourage you to take a look at the National Academy of Medicine report that came out on meaningful engagement of communities. You cannot meaningfully engage communities without understanding the equity imbalances. And by the way, I'll stop now, but please keep in mind that there are also equity imbalances between those of us at NIH and many of you on the ground doing the work. And we also need to support research that helps us make progress. The Fogarty International Center has started that effort, and we hope that when we talk of real equity, it wouldn't just be between urban and rural and different tribes and socioeconomic, but it would also be the equity imbalance between the funder of the research, sometimes the US investigators and partners in low and middle income countries. This is a wonderful time for us, and I think all of us should participate in making sure when we talk equity, we're talking very broadly and we can make progress.
Thank you very much. About equity, can I ask you something about the inequity between sickle cell patients who are living in urban areas and those, majority of them are living in the rural areas? So my question concerns also the use of the so-called traditional medicine in sickle cell disease. I mean to talk about the ethno-medicine. What is your feeling about it?
I cannot cite a specific NIH position on this, and so don't take my response to represent an NIH position. There's no question at all that there may be real value in certain ethno-specific strategies. In fact, many of the herbals that my great-grandmother and my grandmother used were effective. Now, it is very possible that if we invested enough resources, even in the private sector, in pursuit of what are the essential ingredients in those that lead to efficacy, I think we should take the same traditional framework. Is there efficacy demonstrated and then effectiveness? But the one thing that we sometimes forgot, that as I've become older and perhaps a little wiser, I know that part of the reason why my grandmother's prescriptions worked was not so much what she was given, but the trust that the people had in her, that that woman had that patient or that community's health in mind. We are lucky that as physicians, we are still trusted, but we can lose that trust very quickly. And so we shouldn't separate the equity, the ethnic, cultural potential therapies from the central issue of trust, and then approach it in a scientific manner, but not poo-poo it as it's not biomedical research and therefore cannot be trusted. I think there's a lot we can learn from China in what they've done in their primary health care, where sometimes actually the pharmacy would have a section on traditional approaches as well as the current day drugs. So I think there's a lot of work that needs to be done in that area.
Thank you very much. Any other questions? Because, yeah, please.
Thank you, Dr. Mensah, for this question. it's a lot. We have a lot of bench result, a lot of things which we know really is working. For me, what is really a barrier for moving when in implemented side? Is it because there is no more specialists to design this kind of study, or there is those kind of study are really expensive to really put in place? Because many evidence are now shown. For example, newborn screening, many evidence are not shown. But why cannot move in implemented side? Is it because of there is no more specialists to really design this kind of study, or is it really expensive to do this kind of study? Thank you. This is Dr. Saro from Mali.
Dr. Saro from Mali, thank you very much for your question. Dr. Saro, I'm surprised when you got the microphone, I thought you were going to ask me about Mansa Musa. And we're going to ask whether I'm related to Mansa Musa. But actually, my last name is a little different. And unfortunately, my family is not related to the riches and the wealth of Mansa Musa. I think your question is very important. And there are a few things we already know. It is not the number of specialists. In fact, for many of these studies, if you have invested, trusted, experienced individuals, whether they're nurses, whether they're pharmacists, whether they're physician assistants, whether they're community health workers or social workers, it really doesn't make any difference. You really don't need the cardiologist or the neuroscience specialist to make these effective. I think what it takes is beginning with that meaningful engagement of the particular sickle cell disease community you're working with. What is the essential need they have? The mistake we've made in the past decades is we come as, I'm George Mensah, I'm from the NIH, and our strategic vision and strategic priorities identify these 5 areas, and we'd like you to apply the research in those 5 areas. And that's people go where the money is, and that's what they do. We're beginning to turn this around. It has to begin with community engagement and identifying the individuals. They may not be experts. You may actually need a pastor or a traditional healer on your research team or advisory group. But it begins with using frameworks that have been validated, that have been shown to either address adoption or address sustainability or address fidelity, and design your studies in that way. If you give me, I'm a cardiologist by training, and most of the research that I worked on had to do with biological endpoints. If you give me antihypertensive and you tell me you want a 5 millimeter reduction in blood pressure, I can tell you within 5 minutes the sample size you need and the study design that you need to do that NIH will get excited about. It's more challenging when we're thinking of, if we're going to study adoption, sustained adoption, and we're looking at the proportion of people who would stay on hydroxyurea for at least 4 years. How do you design the cluster size or the registries that you use in a randomized registry study? We need biostatisticians for sure. We need design people for sure. But the last thing we need is the super specialized cardiologist or neurologist or surgeon. I don't see that as really critical, but we need trusted, committed community health workers, social workers, nurses, and general physicians. And of course, if we have access to a super hematologist like Ambrose, that's great. But Ambrose, I'm sorry, I don't think you are absolutely essential in making sure that we make progress in this research.
I agree, George, but I'm actually a geneticist, not a hematologist.
I stand corrected. I stand corrected. Ambrose is so talented, I sometimes confuse him for a cardiologist. When I met him with Bongani Mayosi, I thought he was. But truthfully, we need your help. The time has come to move away from the rigorously designed controlled studies that have so little external generalizability that when we get to the street level on the day-to-day use, we see serious limitations. In fact, for me, that is the real excitement I see in your registries. But please, let's not support this registry work for 10 years. And then after 10 years, that's when we take all the lessons and say, here are the great findings of 10 years of work in Sickle in Africa, or the global approach. Let's do this right from the beginning. As you're designing your registries, how are we thinking about the implementation challenges? How are we thinking of literacy challenges? How are we thinking of the trust issues that people in the rural areas may have about those city professors? So we have a lot of work to do, but I'm very excited and very, very optimistic that you are the people who are going to lead us forward in this area.
Thank you very much. If you have some minutes more for us, we have another question.
Merci. Je ne sais pas si le professeur peut m'entendre en français.
Un petit peu, je l'ai étudié au lycée mais il y a longtemps, 1969. Donc, je ne parle qu'un très petit peu. So please have mercy on me.
OK. 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 Moi, c'est Dr Assadjara. Je suis patiente drépanocytaire. Je suis du Mali. J'ai vraiment apprécié votre présentation. Vous avez pris en compte beaucoup d'aspects par rapport à la prise en charge de la maladie, l'implication des patients, l'utilisation de résultats. Mais la question que j'aimerais peut-être vous demander, est-ce que vous connaissez un des pays qui a eu vraiment de l'expérience par rapport à l'utilisation de ces résultats pour améliorer la qualité de la prise en charge des patients drépanocytaires? Ça, c'est la première question. La deuxième question, depuis ce matin, il y a beaucoup de pays qui ont parlé de l'utilisation de l'hydroxyurée dans le traitement de la prise en charge du patient. Mais est-ce que vous, en tant que spécialiste, est-ce qu'il y a des études qui montrent l'impact sur les effets secondaires de l'hydroxyurée sur l'utilisation à long terme chez les patients drépanocytaires? Donc, ça, c'est les deux questions qui me tiennent à cœur. Merci beaucoup.
Thank you.
Merci beaucoup, Madame Professor, but I would need some help. I got the general sense of your questions, but maybe if someone, I know there were two questions, if someone could translate briefly the first question for me, and I'll try to answer. I wish I could speak French, but my French is really horrible.
Okay, for her first question, she would like to know, based on what you present today, and she really see a good of research, which when it's translated, people will benefit about it. And she would like to know, is there a country in the world who really is good on this translation, who take those basic sense from bench coming on the patient side? And is there a country who did very good on that? If there is this country, she would like to have an example of that. Thank you.
It's an excellent question. And unfortunately, it's difficult to answer specifically. First of all, as you know, implementation science is relatively new. And so we don't have a good base of data to make fair comparisons. But I can tell you where they're at least making greater strides and real progress. I will cite Canada as one example. Even how they have approached the fundamental discoveries and the knowledge they know about which drugs work in high blood pressure control, and how they've managed to get a much higher blood pressure control level than the United States. So I wish I could say the United States is an excellent example. I cannot. And unfortunately, I don't think there's good data to say definitively which countries are better. But if you send me an email, I will work on it, and I can get you at least 3 to 5 countries where we have very, very good evidence. By the way, the World Health Organization has a section that focuses on implementation science. So they may have some evidence of where there are either a lot more people supported on this, but the truth is all of us can do very, very good job. And I wish in 5 years I can say that Mali or Ghana or Cameroon or Nigeria, and that we also have examples of where we've taken this to test. So excellent question. Don't have a definitive one country, but when I think of blood pressure, for sure, Canada is doing better than the United States based on how they're implementing proven effective interventions.
Thank you, Professor. The second question concerns hydroxyurea for a long time. She asked you if you know all the side effects in patients who use hydroxyurea for a very, very long time.
I wish my colleague, Dr. Panepinto, was here, but honestly, many of you may be the experts that I should consult. As I told you, I'm a cardiologist, so I haven't personally had even more than a few patients who've been on hydroxyurea. But that question can be answered. I think sometimes, though, we focus on the side effects and scare people off. And many patients continue to suffer painful crises, acute chest syndromes, when the benefits actually outweigh the adverse events. But the question of very long-term therapy, we have good evidence over more than 10-year use, and I think it would be good, even in your registries. These are the type of studies that I'm hoping you'll be able to publish and inform us. As I remember reading the paper on the registry, the Sickle in Africa registry in Kumasi. They already identified what were some of the most common symptoms and adverse events, but having a long-term database would be very helpful. I would be happy to look into that and consult some of my colleagues here at NIH and be able to share that results with you.
Thank you very much. We want to thank you again for this fantastic key talk, and we clap our hands again. Thank you very much.
Thank you.
Thank you.
Thank you, Wanda, for having me.
Okay. Thank you. Yes. Thank you. We have 10 minutes more. If you want, we can have a short discussion about what we have learned today. Many lessons, many good things before lunch. Oh, Ambrose, have you something?
No, it's something funner than discussion. I think we haven't introduced ourselves, but that's the right time for everybody to introduce itself.
That's a wonderful idea.
So the mic has fallen to Elijah or Emmanuel. My name is Emmanuel Balandi. I'm PI for SPACO Tanzania. Dr. Mensah, thank you very much for your talk.
Thank you.
Thank you very much, colleagues who are here, for a wonderful, great meeting that we are having. Thanks very much.
Thank you.
Okay, so I am the sole NIH representative on site for this consortium meeting, and thank you so much for having me here and hosting this meeting. My name is Andrew Loudon. I am a program director, program officer at NHLBI. I have been here for at NHLBI for 2 months, almost to the day. It's been a fantastic experience so far, and it's a pleasure to put names to faces. Thank you again.
And I'm delighted to say that when Andrew joined NHLBI, I met him the first day in the hallway and we hit it off. So I'm looking forward to continue our dialogue. Delighted to see you, Andrew.
I'm Joseph Rujumba. I'm a senior lecturer from Makerere University, and really my focus is on behavioral science and qualitative research. I'm a member of the Sparkle team in Uganda. Thank you.
Excellent.
I'm Ruth Namazi from Uganda. I'm a pediatrician and a lecturer at Makerere University, and I'm a pediatric hematologist as well.
Great.
Good day, everyone. I'm Andre Pascal Tingni. My background is further internal medicine and AP, and I'm a member of SPEDEC based in Cape Town, South Africa. Thank you.
Andre, I remember when we first met in Cape Town, so delighted to see you again, Andre.
Hello, everyone. I'm called Anana Mazi, and I'm the coordinator for Sparkle Uganda.
Great. Thank you.
Hello, everyone. Thank you, Professor Mensah, for that great talk. My name is Patience Kuona. I'm a pediatric hemato-oncologist from Zimbabwe and the principal investigator for the Zimbabwe-Zambia site. Thank you.
Great, thank you.
Thank you, Professor Mensah. And hello, everybody. My name is Gwen Kandawashika. I'm a pediatric neurologist working together with patients. I'm co-PI. Happy to be here.
Great, thank you.
Thank you, Professor Mensah, for the quite elaborate and lovely talk. My name is Anaze Zemadu. I'm from the Nigerian site and the malaria site lead. I'm an adult hematologist.
Great, thank you.
Thank you so much, Professor Mensah, for a great talk, very informative. My name is Diana Incha. I'm a senior lecturer from the Department of Biochemistry and Molecular Biology. I'm also a PI from the Clinical Coordinating Center, CCC. Thank you.
Great, thank you.
My name is Lulu Chirande from Sparkle, Tanzania, and in Sparkle, I'm involved with the newborn screening implementation and research. Thank you very much for the talk on implementation science. I'm a pediatric hematologist.
Great, thank you.
Thank you so much, Prof, for a great presentation. My name is Irene Minja. I'm a co-PI at the Sparkle Clinical Coordinating Center and also skills development lead, technical lead at the Sickle Cell, the CCC and Sparkle Tanzania. Thank you.
Right, thank you.
Thank you. My name is Agnes Jonathan. I'm a management specialist with expertise in public health and business management. I'm project manager by QnA Institute. I'm a program manager for CCC, and I also work with Sparkle Tanzania. Thank you.
Great, thank you.
So, hi, everybody. My name is Upendo Masam. I'm research and data coordinator for Clinical Coordinating Center, but also I'm a statistician.
Excellent, thank you.
Good morning, everyone. I am Sickle QnA, clinical hematologist. I am the focal person of malaria chemoprophylaxis protocol from Sparkle Mali team. Thank you.
You.
Hello, Professor Mensah. Thank you so much for a great presentation. And I'm Abdul Malik Koya, fully supported SEDEC Research Fellow for the Mali site. And I would like to disappoint you a little bit because I'm trying to embark on a super specialty, a little bit of you, the cardiology and genetics, to look at the
implications of cardiovascular science on sickle cell disease. I think that will really annoy you a little bit, but I'm very sorry.
You'll be pleased to know I'm not disappointed at all. And in fact, every opportunity we get, we have a responsibility to support your training and career development. I think the general message is we shouldn't rely on just the tiny little few specialists we have to enable us to do the work that needs to be done. And we should think of ways to use technology so that the few specialists we have can be made accessible very broadly. So please keep up the great work, and I look forward to hearing from you if there's anything we can do to support your training and career development.
Okay, c'est Touré Bouakar Ali. I'm a hematologist and sparkomality.
Merci beaucoup.
Jamwali, bonjour. My name is Hans Ackerman. I'm a physician scientist in the intramural program of the NIH. I spent 3 years at the National Heart, Lung, and Blood Institute. Before that, I was a fellow in the hematology branch and the sickle cell clinic and in the critical care medicine division. I lead a intramural research program now at the Institute of Allergy and Infectious Diseases. And like Abdul Malik, who I just met, I'm focused on a very singular signaling mechanism in the human vasculature, looking at the role of hemoglobin as a regulator of nitric oxide signaling in the endothelial cell. So, Professor Mensah, this is aim one of the NHLBI priorities: understand normal function in humans. But of course, my interests go broader. I'm interested in malaria, sickle cell disease, and that's why I work closely with Professor Gindo and the center here in Mali, where our institute has been invested for more than 30 years now. So it's a great privilege to be here with you in person today.
Excellent, Hans. And maybe let me use Hans's introduction to make the point that unless we have fundamental discoveries and have a better sense of normal biology, there will be nothing to implement and there'll be nothing to translate. So please understand that all aspects of the translational pathway are very important. The message is that traditionally we have focused so much on the great science and the discovery, and then we assume that it would happen. I think it's wonderful to have you there, Hans. It's wonderful to have Andrew. This is the time for us to have that strategic partnership that involves basic scientists, social scientists, implementation scientists, so that we can cut down how long it takes to get the discoveries into real world impact. It's unfortunate that we have to come to Bamako for Hans, for you and I to connect with Andrew, but I think that's why I said, even for us at NIH, there is a message for us, and we need to hear from you how we can do better. Looking forward to connecting with you in the intramural when you're back here.
Thank you, Prof. I am Altuma Gindo, hematologist, and I'm the director of Sickle Cell Disease Research and Control Center of Bamako. And I am also the SPARCO PI in Mali.
Wonderful, thank you.
Thank you, Prof. Mensah, and good afternoon, everyone. Namush Jombo is my name. I'm a pediatric infectious disease specialist from the University of Zambia. I head the Department of Pediatrics and Child Health. My interest is in infectious diseases, and of course, infectious disease cause of patients being the leading cause of mortality. And also, I think my concern is also among some of the interventions that we mentioned. I didn't hear anything about immunizations, which we think I think is a very critical component in this course of patients. I'm quite happy. I've seen the penicillin prophylactic immunization is quite critical, and much of the vaccines that we need in this course of patients are actually not provided in the routine EPI program. And actually, that will be an opportunity also to catch especially adults. And then thank you so much for your presentation on implementation science. I think that's the biggest missing link in most of the activities that we do. For instance, we could be celebrating our milestones quite prematurely because we are looking at the actual numbers, not looking at the actual impact. For instance, one center shared something on 70% hydroxyurea uptake at the facility. What does that really translate in terms of impact? For instance, if that center has got 1,000 patients, you're actually just capturing 70% of the 1,000. How does that 1,000 relate to the actual population? Maybe the actual population of patients is 50,000. You're catering for 1,000 and you have 70% uptake. Even that 70%, probably another 50% is dosed appropriately. At the end, you have 300 patients on effective dosing of hydroxyurea. How does that give you the impact that you need? So it could really be celebrating prematurely if we leave out the component of implementation science. Thank you very much.
That's fantastic. And that's also another example where the equity implementation framework can be used to address the reach of this intervention that we know to be effective. So I'm really, honestly, I'm very, very excited about this consortium and the transformative work you can do, especially if we are truly committed to working across disciplines and bringing in biostatisticians, disease modelers, implementation scientists, as well as basic scientists. So great expectations of what you are going to help us accomplish. And I hope I can join you in person next year. But anyway, this is really great. Please continue.
Good afternoon, everyone. My name is Kuthala Mniga. I'm from South Africa. I'm a lecturer in the University of Cape Town. But today, I'm just happy to introduce myself as a PI for the inherited kidney disease using whole exome sequencing. It shows growth. Professor Ambrose mentioned earlier that people, they make sure that they grow to an extent. So I'm just happy to announce that I have my own grant working on inherited kidney disease as a young researcher.
That's fantastic. That's fantastic. Thanks for sharing that.
Thank you so much, Prof. My name is Bruno Mbando. I work with Sickle, Tanzania, and I'm a biostatistician.
Fantastic. And thank you for moderating that session with Emmanuel last year.
Thank you, Prof. I'm Daniel Kandonga from Sickle, Tanzania. I work as database coordinator and also I'm a developer of biogas and for sickle cell disease in Tanzania. I have a background in computer science and health informatics. Thank you.
Great. Thank you.
Hello, Professor Mensah. Hello, everyone. My name is Rafael Sangeda from Tanzania, Sickle CCC. With interest in data and epidemiology, and I can't wait to embrace implementation science.
That's fantastic. And I hope those of you with interest in data science, that you're keeping track of the Fogarty funding for data science innovation in Africa, DSI Africa. NHLBI is a co-funder. We funded several grants under there, and we're very, very excited about that program. Any opportunity you can get to use data science to advance the care of sickle cell disease would be something that we'll be very, very interested in.
Thank you very much, Prof, for that very insightful and stimulating talk you gave on implementation and research. I'm Catherine Chundalioka. I'm a pediatrician and also a lecturer in Zambia. And I'm happy to report that Zambia, in collaboration with Nigeria and Ghana, are actually doing implementation and research with a UK-sponsored organization where we are trying to understand how we can improve care of sickle cell disease patients, hearing it from the patients, from the caregivers, as well as the health workers. Thank you.
That's fantastic. I know there is one study, I'm blanking on the name of the PI, but that was done at the Kolibu Teaching Hospital that demonstrated almost a 90% reduction in maternal hemorrhage-related fatalities. Just imagine we were able to identify the primary drivers of such success and be able to scale up and spread to other parts of those countries. So please keep in mind, we are very, very interested in supporting investigator-initiated research in sickle cell disease that really focus on dissemination and implementation research. If you're not sure what you're doing falls and aligns with our NHLBI mission, you have Andrew there, or drop me an email and we'll connect you with the right people in the extramural world. So please make that a priority. And the individual work that you all do would also really enhance the collective registry work that Sickle in Africa does. So thank you for mentioning that.
Good afternoon, everyone. My name is Cynthia Piri. I'm a social scientist, and I'm from Sparkle, Zimzam. Thank you.
Thank you. Thank you.
Good afternoon, everyone, and thank you, Prof, for your enlightening presentation on implementation science. My name is Natasha Kaweme, and I'm the research fellow from Sparkle, Zimzam, Zimbabwe, Zambia.
Great, thank you.
Good afternoon, everyone. My name is Josephine Guy from Sparkle CCC, coordinating standards of care activities as well as skills development activities for the consortium. I'm also the laboratory scientist for the sickle cell disease laboratory in Tanzania. Thank you.
Excellent, thank you.
Good afternoon, everyone. So my name is Janet Manongi. I'm the head of operations and also sustainability coordinator for this Sparkle CCC.
Great, thank you.
Good afternoon, everyone. My name is Rita Mutagonda. I'm a clinical pharmacologist with a pharmacy background. I'm also a senior lecturer at the Department of Clinical Pharmacy and Pharmacology. At the consortium side, I am the co-principal investigator in malaria chemoprophylaxis study. Thank you.
Thank you.
Hello, everyone. I'm Chandra Osterwey-Kliu. I am a lab technician as well as coordinator for SADA.
Thank you.
Morning, Professor. My name is Mutaka Jallo. I'm a IT manager of Mumbai.
And thanks a lot for your help. I'm delighted I was able to give my presentation and have a chance to join you. I know I'm standing between you and lunch, and so I'm happy to stay, but please, whenever the time is appropriate for you to stop so you can get some nourishment, you could do so. But thanks a lot for keeping.
Okay, thank you. Good afternoon, everyone. My name is Xavier Chinge from Zimbabwe, Zambia, responsible for data management activities between the two sites. Thank you.
Thank you.
And thank you, Prof. Good afternoon. My name is Ian Rurezo. I am a medical lab scientist by training and investigator in the SHAS from the Zimbabwe site. I also have a keen interest in implementation science in terms of health system strengthening. And thank you very much for that talk. Thank you.
Thank you.
Thank you very much, Dr. Mensah. My name is Maxwell Mwingu. I'm from Nigeria, Wako, Nigeria. I'm a chemical pathologist by training, and I oversee the laboratory for sickle cell newborn screening at the University of Abuja. I'm also a co-investigator on the train grants.
Excellent. The train grant is one of our capacity building research efforts that we've supported in the last decade. So thank you very much for mentioning that.
Thank you very much, Prof. Good afternoon, everyone. My name is Ruben Chanumba. I'm a biochemist by training, and then I support the Nigerian data management. Thank you.
Thank you.
Good afternoon, everyone. My name is Patrick Moore. I'm the SPACO Fellow for Nigeria. Thank you.
Thank you.
Good afternoon, everyone. Afternoon, Prof. My name is Takuto Tisi. I'm from Zimbabwe, and I coordinate the Zimbabwe and Zambia sites. I'm probably the most excited person in the room because of your talk. I'm a lab scientist by training, so I come from basic sciences, but I've spent the last 10 or so years working in implementation science, and I'm actually trying to work on a K43 this year in translational research and implementation science. Thank you.
That's fantastic. Thank you.
Good afternoon, everyone. Thank you, Prof. My name is Onyie Udozo. I'm a computer engineer by training, and I'm from SPACO, Nigeria. Thank you.
Thank you.
Hello, everyone. My name is Yajim Prof Mensah. I'm a pediatrician, and I'm a heme oncologist in training. I'm with SPACO Ghana, and I'm actively involved in the newborn screening at the site. Thank you.
Excellent. And she didn't tell you, by full disclosure, is this my sister? So I was just kidding. Yes.
Good afternoon, everybody. Blessing Inegbejo, representing SPACO Nigeria. I'm a member of the PPIR group and also a coordinator in the University of Abuja Teaching Hospital Patients Working Group. Thank you.
Thank you.
Hi, Professor Mensah. Thank you for your talk. My name is Valentina Gobitungi. I'm from Cameroon. I'm here as part of SADA, working on the biorepository group. And I'm proud to say that I'm the first student of Ambrose, the first PhD student. I always say that. I'm planning to do implementation studies. So I think I will, I don't know if, I don't know, yes. So I'm sure I will share some mail with you. Thank you very much.
Excellent. Thank you very much.
Bonjour, professeur. Moi, c'est Khadija Barego, malienne, ici de l'épanouisseur, et membre de l'Association malienne de la lutte contre la tuberculose. En lutte.
Thank you.
OK. Je pense que je me suis déjà présentée. C'est Dr. Jara Assa. Je suis membre de l'Association malienne de lutte contre la drépanocytose. Et je suis aussi chargée de recherche à l'Institut national de santé publique au Mali. Je souhaiterais vraiment continuer le contact avec le professeur s'il est disponible pour approfondir mes connaissances par rapport à cette pathologie. Merci.
Merci beaucoup.
Thank you, Professor. My name is Fatmata Tugula, part of SPACO Mali, a member of Association Malienne de lutte contre la drépanocytose. Merci pour thank you for your presentation and your availability. Thank you.
Merci beaucoup.
Hi, everyone. My name is Lawrence Odeditu. I work with here in Kumasi. I'm a pediatrician, pathology and oncology.
Thanks, Prof. Jingo. I am faculty at Makerere University, but I also am a member of the Uganda SPACO team, where I lead the registry implementation. I am a bioinformatician by training.
Excellent. Thank you.
Hi, my name is Vicky Nembaware. I am the project manager for the SADC grant, and I'm also a senior lecturer at the University of Cape Town. Thank you, Dr. Mensah, for giving up your holiday to be with us. And just to confirm that we are visiting you in August next year. So we'll send a calendar invite. No need to worry about traveling. And I'm also a co-course convener for the Data Science Masters at the University of Cape Town, which was launched this year. Just throwing it out to the consortium in case we can come up with some days for that. Yeah, thank you.
That's fantastic. Vicky, thanks for all your help. I almost got the time wrong. And so in the middle of the night, I emailed Vicky and Julie and Vicky responded and got me all straightened. So really appreciate it.
Yes, my name is Ivan Swasamuzu, originally a biological scientist. Now as a cameraman doing the social media. A fellow. So as a fellow, I'm working on hydroxyurea adherence, looking at what are the adherence rates, and then as a master's thesis to evaluating the Hughes scale for hydroxyurea adherence. Thank you.
Excellent. Thank you.
Thank you. I'm Dr. Saro Yeya, SPARC coordinator from Mali. And it was really great to hear about this topic because in our Master of Public Health program, we just enrolled the first cohort of implemented science because now it was 8 million, but the first cohort of implemented science will graduate next year. And just to confirm that your hope that we will move forward soon. Thank you.
Excellent. Thank you.
My name is Mario, so I'm responsible for the infrastructure for SEDEC. Thank you.
Thank you very much for your help.
Thank you very much, Professor Mensah. I'm Leon Chilolo. I have the chance to be the co-chair of this fantastic consortium. Now I would like just to wish to all the participants bon appétit. Goodbye. Thank you very much for your ability.
Bon appétit, mesdames et messieurs.
Merci. Très bien, nous faisons la pause pour le repas. Merci beaucoup pour cette session qui a été très intéressante. Nous reprenons dans 1 heure. Merci. Bon appétit.
Bye, everyone.